Hello everyone, thank you for coming to Applied Cells tutorial. My name is Liping Yu. I'm the VP of Application at Applied Cells. It's my pleasure today to present to you centrifuge-free, matrix-free cell isolation found on Mars. Thanks to the CYTO community, Mars was the winner of Innovation Showcase in CYTO 2020. We have continued to develop the technologies and applications on Mars platform. Today, I'm going to give you the update and show a few applications. Life science research involves processing complex samples that contain heterogeneous cell populations. The advancement in fields of multi-omics analysis and cell-based therapy enable deep analysis and best use of cells. However, quality of the analysis and therapeutic use of cells depends on efficient cell isolation and purification processes. Conventional tools are limited in automation, cell recovery, and capacity. There is always trade-off between different desired features. How to minimize the trade-offs and get an overall satisfaction in cell processing and cell isolation has been the challenge for scientists and engineers. Here is our solution. We have developed a novel acoustic cell processing chip and novel magnetic cell separation technologies. The acoustic chip is able to separate cells based on their physical parameters and move cells into fresh buffer, so functionally we use it to replace centrifuge. The Mars magnetic separator is an inflow matrix-free separation channel, and it automatically recovers both positive and negative portion of cells. We build each of these technologies into an independent module and put them into different configurations. Because Mars process involves little human operation and takes fewer steps than conventional process, it has the feature of high recovery, high purity, high reproducibility, automation, and less stress to cells. Let's review how we achieve size-based selection of cells on the acoustic chip. When a mix of beads come into the chip channel as a laminar flow, under the influence of acoustic wave, beads at all sizes will tend to move toward to the center of the channel, but larger beads move faster than smaller beads. This way, the larger beads will end up being collected in the outlet at the center of the channel, and the smaller beads will be collected in an outlet at the side of the channel. By increasing the acoustic driving strength, all beads move to center faster, so more smaller beads can be collected at the center outlet at a certain amplitude. Doing the amplitude sweeping, we can find a threshold value where different sized particles are being separated. For example, let's look at amplitude 13. Here, 60% of the 5 micron beads are being collected in the center. 3 micron, 2 micron, and 1 micron beads have a lower than 10% recovery in this outlet. This way, any beads that's larger than 5.6 micron can be separated from the smaller beads. And then move to 17. Here, all 3 micron beads are also being collected in the center. So we have a separation of 3 micron beads and 2 micron beads and 1 micron beads. Can we move 2 micron beads and 1 micron beads to the center outlet? The answer is yes. On Mars acoustic chip, there are more parameters to tune. Flow rate is another one. Lowering flow rate allows beads to be recovered in the center outlet at a lower amplitude. That is to say, if we can take a slower flow rate, we don't have to drive the chip so hard. We can even recover 60% of 2 micron beads at a 0.4 flow rate at a 22 amplitude. On Mars, you can tune a condition that best fits your samples. How can we relate the beads selection on acoustic chip to cells? So in the application of isolating leukocyte from lysed whole blood, we discovered a correlation of beads to blood cells and cellular debris. This plot shows the established correlation. By comparing the driving amplitude and the recovery of beads and cells, we find that 3.4 micron beads behavior in acoustic field is quite similar to leukocyte, and 1 micron beads behaves similarly to RBC lysate. So with the beads, and we could optimize a condition that reaches the highest cell recovery and the lowest debris recovery. In this application, the blood to RBC lysis buffer ratio also plays a role. The lower lysis buffer leads to higher cell recovery, but less debris removal. We have tested leukocyte isolation from samples of different donors, different volume, on different modules, and achieved 90% cell recovery in all conditions. The result demonstrated that Mars CPC process is a donor-independent, volume-independent, and module-independent process. This is another application using CPC to select cells based on size. It is a label-free, centrifuge-free tumor-infiltrating lymphocyte isolation process. A human lung tumor biopsy sample was enzymatically digested. The cell suspension was processed on CPC twice. First time, at a relatively lower amplitude, the larger cells were mostly enriched in the center outlet, and most of the TILs and debris are collected in the side outlet. Second time, we run TIL and debris portions through CPC at a higher amplitude. Then TILs are isolated from debris and collected through the center outlet. Now enriched TILs has much higher purity than the original sample and is ready for downstream analysis or culture. Let's look at some applications where both CPC and MAG separation are used. In a collaboration with Penn Medicine, we support the group to develop a new disseminated tumor cell isolation and analysis method. DTCs are very rare in patients' bone marrows, and the conventional IHC method to analyze them takes a long day of work. Mars workflow is described here. We spike human breast cancer cell line cells into healthy donors' bone marrow, and we label the cells with anti-CD66B magnetic beads. That is for granulocyte depletion. And then we do RBC lysis. And now the sample is ready to go on Mars. On Mars, they run a C1, C2, M1 serial program. The program is to wash leukocyte bone marrow cells from the lysed RBC and to concentrate bone marrow cells and tumor cells, and then do magnetic depletion of granulocyte. The enriched DTCs are ready for staining with antibody panels for flow analysis. And then the stained DTCs are going through CPC again to wash off free antibodies. And then it follows by flow cytometer analysis. The entire process takes less than 1.5 hours, so it's a much faster process. In those experiments, tumor cells were spiked in bone marrow cells at a frequency of 1 in 100,000 to 1 in 10 million bone marrow cells. And you can tell overall 80% recovery of tumor cells were achieved on Mars. In comparison, in the IHC processes, cells were spiked in at 1 in 10,000 to 1 in 100,000 kind of frequency. However, a lower than 60% recovery were achieved. So this shows that using Mars and a flow cytometer could make a much faster, potentially more sensitive method to DTC detection. Another example using both CPC and MAG selection is granulocyte isolation. Granulocytes are easy to be activated during cell processing. We assess the activation status of neutrophils by measuring the shedding of CD62 ligand. Using diluted whole blood and lysed whole blood as a control, we found that processing 1 ml whole blood on acoustic chip that takes 5 minutes, and processing 10 ml of whole blood on acoustic chip that takes 15 minutes didn't have a significant impact to neutrophil compared to conventional FICOL-based separation processes. That processes takes over an hour to complete. So with this result, we think Mars CPC chip cell washing can be a more gentle processes than standard FICOL separation. Taking advantage of a gentle washing process on Mars system, we have developed a two-step enrichment workflow to isolate granulocyte from human peripheral whole blood samples. Because granulocytes are larger than monocyte and lymphocyte, we apply an amplitude on the acoustic chip that enrich granulocyte over other cells and debris. After this, and we also use magnetic beads that target non-granulocyte cells to deplete those cells on the acoustic channel. So combining the two processes, we were able to get 99% purity of the isolated granulocyte. The previous two examples are about negative enrichment. Next, let's look at positive selection on Mars. Since Mars is a matrix-free channel and has self-rinsing and releasing functions, Mars magnetic separation has the feature to do positive selection of immune cells from no-lyse, no-wash samples, and that applicable to peripheral whole blood, bone marrow, and apheresis product. Mars positive selection doesn't require a PBMC preparation that a conventional protocol requires. So Mars selection has fewer steps and a much faster speed. Here is an example of CD3 positive selection from leukapheresis cells. In a leukapheresis bag, CD3 cells starts at 73% of the entire leukocyte population. And after my selection, T cell purity got over 98% and viability over 90%. We take the isolated CD3 T cells and use CD3 and CD8 to activate the T cells. After continuous culturing for 7 days, the activated T cells are still over 98% purity and high viability, and it express expected CD4 and H distributions, and the cell populations dominated in the activated cells are memory T cells, central memory T cells, and they are featuring CD45RA negative and CD62 ligand positive. You may want to ask how we complete 1 billion cell isolation on Mars. Mars magnetic separator is a scalable technology. We could use 1 MAG module or 3 MAG modules. This table shows you how we process millions of cells to 1 billion cells on Mars. The purity and the separation efficiency remain the same at different scales. To process 1 billion cells, we run 3 modules in parallel, and each module has releasing cycles. When the channel reaches its capacity, the positively captured cells are released from the channel, and the channel is ready for next cycle. It only takes 2 cycles and 35 minutes to process 1 billion cells. To summarize my presentation, on Mars platform, we can select 1 target cell out of 1 million cells and select target cells out of over 10 billion cells or anywhere in between. Just choose the one that fits your need. you very much for your attention. Now we are ready for your questions.
Thank you, Dr. Yu, for the update with examples of applications we have developed at Applied Cells. As mentioned, Applied Cells has combined two powerful technologies into a single solution for a complete automated workflow performing cell washing, cell concentration, and immunomagnetic isolation using both positive and negative techniques. Our technology doesn't just simplify the sample preparation process. It's designed to save time and resources without compromising on high purity, high recovery, and high reproducibility. We can achieve this by using a matrix-free, centrifuge-free, and in certain applications, label-free. Matrix-free means no magnetic columns containing iron balls are needed to enable high-performing magnetic separation. This minimizes areas in which cells, such as both target and non-target cells of interest, are trapped, reducing recovery and purity. Centrifuge-free means we do not require the multiple centrifugation steps in such processes as phycol to isolate white blood cells of interest. MARS provides a gentle process without pelleting the cells that can cause cell aggregation and activation. And in some applications, we provide cell enrichment that is a label-free approach, leaving precious cells unlabeled, unaltered. As Dr. Yu mentioned, we are able to enrich TILs and tumor cells by simply using gentle acoustic waves on a lung tumor tissue sample. In summary, by combining this matrix-free, centrifuge-free, and in some cases label-free, we provide a gentle, high recovery, high purity, and fully automated, intuitive solution to sample preparation on samples from a few microliters to hundreds of milliliters of samples. This can be done inside a safety hood, as our MARS system also has a small footprint. Using these benefits, we are focused on three application areas in expanding our solutions. The first is in the genomic solution. We begin with an easy sample preparation that is centrifuge-free with high debris removal without causing cell aggregation. This process is a gentle process resulting in high viability and recovery. Enrichment of target cells can be performed in a label-free manner using our acoustics technology, and then further enriched using our immunomagnetic solution for higher purity. This can be done in a full automated walk-away system. In this case study, we took a look at how we can improve the long, tedious process of doing phycol separation and immunomagnetic separation techniques currently performed today that results in a high/significant loss of cells. This usually requires the customer to start by using large volumes of cells to compensate for this high loss. On our MARS system, we began with only 6 milliliters of whole blood to isolate CD34 cells. In our assay control, we used CD checks as the first process. And in this process, we can see that the starting CD34 percentage of cells is 0.37% of total white blood cells. Post-magnetic separation on the MARS system, we can enrich this to approximately 25% purity with high recovery of these cells. The next three samples represent samples from healthy donors. The first represents a sample that with a starting CD34 count of 0.09% that we were able to enrich to approximately 9% purity. In the next two samples, from 0.02% starting CD34 cells to 0.06% starting CD34 cells, we're able to enrich to over 10% purity. This allows the user to be able to take the sample and move into a single cell sorting system with a faster time to cell sort to get to the highest purity obtainable through single cell sorting. In our next area of focus, tumor biology, we focused on rare cell analysis such as CTCs, DTCs, specifically MRD applications, recovery as low as 1 in 5 million cells in bone marrow. This enables single cell genomics with these few rare tumor cells. Our centrifuge-free approach with minimal number of steps reduces cell loss in sample processing. Large tumor cells are first enriched with using our acoustics by tuning the amplitude to remove the debris and small immune cells of non-interest. Then, through immunomagnetic separation, we can further enrich the target cells of interest. In addition to Dr. Yu's update on our collaboration with University of Pennsylvania's Cancer Biology Group on DTC isolation, I'd like to share with you this case study on plasma B-cell enrichment. Here we have a donor sample in a multiple myeloma study with a low CD138 count. In the top dot plots, you'll see that from the original sample, using CD38 and CD138 positive flow cytometry analysis, we see that the starting material is only at 1.7% of target cells in the bone marrow sample. After magnetic separation using the Mars system, the cells were enriched to 55% purity. This allows the user to be able to do further profiling of these rare plasma cells. And in the third application, we focus on providing solutions in cell therapeutic development. Our focus is providing a highly reproducible, high recovery, and high purity with the option to do bag-in, bag-out capabilities. The integrated sample preparation and cell selection in a matrix-free process reduces dead volume as well as non-target cell trappage. This results in high purity, whether you're doing a positive or a negative selection of the target cells, and high recovery of these target immune cells, which ultimately results in reduced time from vein to vein, and also reduced time in expansion of these cells. It's also a high throughput system in which we provide a 3-module-in-1 approach. This allows the built-in redundancy of these modules, reducing the risk of the loss of samples. For example, for any reason, if one of these modules are not able to process the cells, the remaining 2 modules can continue to process the sample without risking the loss of the full sample. As mentioned, the high reproducibility is of high importance, and this is done through our automation, which also means it's minimal hands-on time as well. The gentle process reduces the risk of causing cell exhaustion. So by reducing the need to do centrifugation and having a simple approach of gently moving the cells through the system without a matrix that could possibly activate these cells, we reduce cell exhaustion. This is also done in a very cost-effective approach because there are no expensive consumables that are required to be able to perform the isolation. From a range of small volume in microliters to large volumes to hundreds of milliliters in complex samples such as whole blood, bone marrow, frozen PBMCs, apheresis samples, and also dissociated solid tumor samples. In this particular case study, I'd like to share with you the results of a negative selection process, and this is done with CD3 positive negative cell isolation from whole blood. In this example, we labeled the whole blood sample with CD14, CD15, CD19, and CD56, which removes the granulocytes, the monocytes, the B cells, and the NK cells. And the remaining T cells are left in the negative fraction. And in this particular sample, we see that we have over 91% purity of the T cells in the negative fraction, with over 95% recovery of these cells. Overall, the Mars enables a wide range of versatile applications, from sample preparation as the front end to a variety of different flow cytometers, imagers, as well as doing rare cell enrichment that feeds into single cell genomics and sequencers to do genetic analysis. And our final application being focused on the immune cell isolation, whether it's focusing on T cells, B cells, NK cells, neutrophils. These are all done easily and gently using our Mars isolation system. In summary, we invite you to join the Mars family, providing for you a sample preparation and cell isolation system in a wide range of capacity that results in high purity, high recovery, and high reproducibility. Hi, everyone. This is Jeanette. Thank you for joining us today. And I was wondering if there are any questions that Liping and I can address for you. I think we have a question on the chat line. The question is, can samples be rerun on the magnetic module in order to increase the CD34 isolation efficiency, for example? The answer to that is yes. You can actually take, whether it's a positive or a negative fraction, and either do a second labeling to see if you can increase the number of magnetic beads per cell, or you could just simply rerun it on the magnetic system. There's no harm that's done to the cells in terms of the rerun. We do, however, suggest that you may want to, the best approach to this is to use another label. If you're looking at CD34, for instance, you may want to use CD133 as another magnetic label to look at a different epitope and increase the magnetic labeling efficiency.
Yeah, I think in this case, definitely, if you rerun the positive collection, and it's highly possible to further increase the purity.
We also want to introduce to you today verbally the fact that we have actually just recently launched an anti-PE magnetic label product. So this gives users a lot more flexibility. So we can either provide for you the PE antibody to go with the reagent, or you can actually provide your own PE labeled antibody of interest and use the anti-PE magnetic particle that we are providing. We've chosen this path because it is highly efficient and really increases the recovery as well as purity using this method. So if you're interested in that, please contact us, and we'd be happy to be able to share much more information and results from our anti-PE product. I think we'll open the Q&A for another minute. If there are no other questions, we'd like to thank you. Liping and I would like to thank you for joining us today. And as mentioned earlier, please feel free to contact us if you have any questions or would like to see a demonstration of the instrument.